Introduction. Glaucoma is a group of eye diseases where nerve cells in the retina, retinal ganglion cells, or RGCs, slowly die, causing vision loss. In most cases, high intraocular pressure, IOP, the fluid pressure inside the eye, is a major risk factor. Treatments currently focus on lowering IOP, but this may not always stop nerve loss. Indeed, some patients continue to worsen despite well-controlled pressure, suggesting other factors are at work. Glaucoma is now understood as a multifactorial optic neuropathy. Age, blood flow, immune signals, cellular stress, and genetics all play roles. In simple terms, glaucoma damages the optic nerve, the bundle of RGC axons connecting the eye to the brain, over time, often starting in midlife or later. While lowering eye pressure is the only proven therapy now, scientists are looking at other pathways because vision loss can continue from aging, reduced blood supply, oxidative damage, inflammation, and other cell-level problems. Plain language summary. Glaucoma is a complex disease. It usually involves high eye pressure, but also aging, blood flow problems, and damage to retinal nerve cells. Treatments lower pressure, but they don't always protect these cells fully. What is GHK-Cu? GHK-Cu stands for a small peptide, 3 amino acids, glycine, histidine, lysine, bound to a copper ion. It is a natural molecule found in the body, in blood plasma and wound fluid. Doctors first discovered GHK in the 1970s as a growth factor in human plasma that could boost tissue repair. GHK-Cu is much studied in dermatology and wound healing. It stimulates collagen and new tissue growth in experiments. Its levels normally decline with age, and people have become interested in it for its anti-aging and repair signals. Overall, GHK-Cu is considered a normal human peptide, often cited as safe and well-tolerated. It can be applied to the skin or taken systemically in research, but there is no approved medical use yet. In this article, systemic effects of GHK-Cu means effects throughout the body, bloodstream organs, not just local skin or eye treatments. Plain language summary. GHK-Cu is a naturally occurring protein fragment that carries copper. It is known to help wounds heal and may influence genes. People study it for anti-aging, but it is not a proven medicine for anything. Overlapping biology of GHK-Cu and glaucoma. Oxidative stress. Oxidative stress is the damage that happens when harmful oxygen molecules, free radicals, build up and overwhelm the body's defenses. It is like cellular rust. High levels of oxidative stress are found in glaucoma and other nerve diseases. Retinal ganglion cells have very high energy needs and rich fatty membranes, making them especially vulnerable to free radicals. Research notes that when oxidative damage occurs, for example, from high pressure or aging, it can trigger inflammation and nerve injury in the optic nerve. GHK-Cu has multiple antioxidant actions in lab studies. In wound experiments, GHK-Cu treatment boosted levels of antioxidant enzymes and molecules like glutathione and vitamin C. It also directly neutralizes toxic lipid byproducts. For example, GHK-Cu can bind and inactivate harmful breakdown products of fats like acrolein and 4-HNE that would otherwise damage cells. In cultured cells, GHK alone, with or without copper, has been shown to reduce reactive oxygen species. Computer analyses suggest GHK-Cu turns on many genes for antioxidant defense. For instance, one review notes GHK-Cu helps support enzymes like superoxide dismutase, SOD, and modulates iron levels to fight oxidative stress. Altogether, these findings suggest that GHK-Cu could, in principle, boost the body's antioxidant responses. However, antioxidant effects in cell or skin models do not guarantee protection of eye nerves. The eye has barriers and specialized chemistry. Simply taking an antioxidant peptide does not automatically cure glaucoma. Also, the body's redox balance is complex. You can't assume more antioxidants always help. For example, some large clinical trials of generic antioxidants in glaucoma have not clearly stopped progression. Summary: GHK-Cu activates many antioxidant pathways and so might, in theory, help cells fight rust. But convincing evidence that it would specifically shield optic nerve cells in glaucoma is lacking. Mitochondrial function. Mitochondria are the cell's energy factories. They use oxygen to produce ATP, the fuel cells need. Neurons like RGCs have huge energy demands, so healthy mitochondria are critical for their survival. Numerous studies link glaucoma to mitochondrial dysfunction. In fact, Glaucoma risk rises with age and with failing mitochondria. Both and RGCs rely heavily on mitochondrial energy. Conditions that hit mitochondria, low oxygen, metabolic stress, can trigger RGC damage in glaucoma. For example, in glaucoma models, high pressure or oxidative stress can impair mitochondrial function in RGCs and even form harmful protein clumps. In human optic nerve diseases like Leber's hereditary optic neuropathy, a pure mitochondrial disorder, only the RGCs die, highlighting this vulnerability. What about GHK-Cu? There's no direct evidence on GHK-Cu and mitochondria in retinal cells. However, we can note some related points. Copper, delivered by GHK-Cu, is a cofactor for key mitochondrial enzymes. In particular, cytochrome C oxidase, complex 4 of the electron transport chain, requires copper. Thus, if GHK safely delivers copper, it might support mitochondrial energy production by supplying this element. But this is purely hypothetical. It's not proven that orally or topically given GHK-Cu ends up in mitochondria of RGCs. Another idea is that by reducing inflammation or oxidative damage, as above, GHK-Cu could indirectly protect mitochondria. For now, this is speculative. We simply don't have experiments showing GHK-Cu restores mitochondrial function in glaucoma. Plain language summary: retinal neurons need a lot of energy. In glaucoma, energy factories, mitochondria, in these cells can fail. GHK-Cu may deliver copper needed by those factories, but nobody knows if it actually helps RGCs make energy. There's no direct proof GHK-Cu fixes mitochondrial issues in glaucoma. Neuroinflammation. Glaucoma is increasingly seen as a brain-like neurodegenerative disease, with chronic inflammation in the retina and optic nerve. When RGCs are stressed or injured by pressure, lack of blood, etc., they release danger signals that activate immune cells, microglia and astrocytes in the eye. This neuroinflammatory response can help at first, but if it goes on too long, it can harm RGCs and neighboring cells. In animal models of glaucoma, blocking certain inflammatory pathways, like IL-1 beta or TNF-α signaling, protects RGCs. Postmortem studies of human glaucoma eyes also show signs of chronic inflammation. Activated inflammasomes and elevated inflammatory markers have been found in the optic nerve and retina. GHK-Cu has reported anti-inflammatory effects in other contexts. Wound studies noted that GHK-Cu treatment not only boosted antioxidants but also dampened inflammation. GHK-Cu and even GHK-Peptide alone can lower pro-inflammatory molecules in skin cells after UV damage and in lung models of smoke injury. In cell studies, GHK orphaned deleterious oxidized lipids and prevented them from triggering inflammation. In plain words, GHK-Cu seems to smooth out overactive immune responses in tissues like skin and lung. But it's a big leap to assume the same would happen in glaucoma. The eye's immune environment is very specialized. We have no experiments on GHK-Cu reducing microglial activation or retinal cytokines. Still, as a hypothesis, if GHK-Cu reduced chronic inflammation systemically, it could help protect nerves. This idea overlaps with general neuroprotection research. Many studies look for anti-inflammatory treatments in glaucoma, but nothing specific links GHK-Cu to ocular neuroinflammation yet. Plain language summary. Chronic inflammation in the eye damages nerve cells in glaucoma. GHK-Cu is known to reduce inflammation in skin and other tissues, so it might help calm the eye's immune response, but this is only speculation because we have no direct data for glaucoma. Copper biology. Copper is a tricky element in biology, essential in trace amounts but toxic if unbalanced. It is an important cofactor for enzymes that protect cells. For example, copper is needed by superoxide dismutase (SOD) and ceruloplasmin, enzymes that break down reactive oxygen species. Copper also helps regulate blood vessel growth and connective tissue enzymes. In fact, a deficiency of copper can impair normal repair and antioxidant defenses. However, free copper ions can trigger more oxidative stress through Fenton chemistry, so the body normally keeps copper tightly bound to carrier proteins. GHK-Cu is interesting because it tightly binds copper in a small peptide complex. In theory, GHK-Cu could deliver copper in a controlled way. For instance, it can reduce damage by limiting iron release from ferritin. Iron can also fuel free radicals. In the lab, GHK-Cu was shown to dramatically keep iron locked up. 87% less iron was released than without GHK-Cu. This suggests GHK-Cu can act like a safe shuttle for metals. On the flip side, taking GHK-Cu systemically could unbalance copper metabolism. If a person already has normal or high copper, extra GHK-Cu might conceivably contribute to copper overload unless other minerals, like zinc, are balanced. For example, people with genetic copper storage disease, Wilson's, get free copper that causes damage. So any use of GHK-Cu needs care. Too much copper, or abusing supplements, can increase oxidative stress. In glaucoma specifically, one study found that altering serum copper-zinc ratios was linked to the disease, implying copper balance matters. GHK-Cu's effect on zinc or liver function is largely unstudied. Plain language summary: copper is needed to help antioxidant enzymes like SOD work, so giving copper via GHK-Cu might support those enzymes, but too much free copper is harmful. It's a delicate balance. Connective tissue and extracellular matrix remodeling. The trabecular meshwork is a spongy tissue in the eye through which fluid drains out. Its state affects eye pressure. In glaucoma, this meshwork often accumulates extracellular matrix, ECM, and collagen, making it less porous and raising IOP. Elsewhere in the eye, connective tissues, like the lamina cribrosa around optic nerve fibers, can also stiffen with age and pressure. In summary, imbalances in collagen and ECM remodeling are part of glaucoma pathology. GHK-Cu is a potent stimulator of ECM assembly, at least in wound contexts. In classic experiments, applying GHK-Cu in rat wounds greatly increased the buildup of collagen and glycosaminoglycans. Gene profiling shows GHK-Cu turns on elements of the TGF-beta pathway, which drives tissue remodeling and fibrosis. In other words, GHK-Cu signals tissues to rebuild and lay down new matrix. This is why it's often promoted for skin and tendon repair. Could this help the eye? One idea is that carefully regulated ECM remodeling in the trabecular meshwork might improve fluid outflow. Since GHK-Cu can change ECM gene programs, it's plausible it might alter trabecular cells. In fact, an independent report, unpublished here, notes GHK-Cu reduced fibronectin and collagen production in cultured trabecular meshwork cells. But those are just cell culture hints. There is no evidence that GHK-Cu lowers IOP or directly remodels eye drainage tissues clinically. In glaucoma, excess remodeling is usually a problem, so any effect of GHK-Cu would have to be precisely balanced. Plain language summary: GHK-Cu strongly promotes tissue remodeling and collagen growth in wounds. In theory, it could also influence the eye's drainage tissue or optic nerve support structure, but whether that would help or hurt glaucoma is entirely unknown. Blood flow and vascular regulation. Normal vision depends on good blood flow to the retina and optic nerve. In some glaucoma patients, especially normal tension glaucoma, poorer blood flow or vascular dysregulation is suspected to contribute to nerve damage. When optic nerve head tissue is inadequately perfused, cells suffer oxygen shortages, hypoxia, and become more vulnerable to injury. Disturbed blood flow and endothelial, vessel lining, dysfunction are active areas of glaucoma research. GHK-Cu has been observed to stimulate new blood vessel formation in wound models. The same lines that boost collagen also produce growth factors that encourage angiogenesis. The MDPI review noted GHK promoted blood vessel growth in healing skin. Thus, one could imagine GHK-Cu might improve microcirculation or repair vessels. There is no data on ocular blood flow from GHK. If systemic GHK-Cu reduced overall inflammation and oxidative stress, that might secondarily benefit vascular health. Plain language summary: good blood supply may help optic nerves survive. GHK-Cu can encourage blood vessel growth in tissue repair. However, we don't know if it can improve blood flow to the eye or protect vessels in glaucoma. Aging and repair signaling. Glaucoma risk rises steeply with age. Many age-related changes in cells, senescence, DNA damage, reduced repair capacity, set the stage for glaucoma. GHK-Cu is sometimes described as a regeneration or anti-aging peptide because it can activate gene programs for repair. For instance, broad gene expression studies show that GHK with copper can shift hundreds of genes toward a more youthful pattern. It can increase collagen and stem the breakdown of tissues in aging skin. Because both GHK-Cu and glaucoma involve age-related processes, some think GHK-Cu might counteract the declining repair signals that accompany aging. The link is highly speculative. If GHK-Cu truly resets cells to a younger state, it could hypothetically make RGCs more resilient. But eye cells are different from skin or lung cells where this has mostly been studied. We also note that GHK-Cu has not been tested in elderly glaucoma models to see if it slows degeneration. Any anti-aging effect of GHK-Cu in humans remains unproven, especially inside the eye. Plain language summary: glaucoma mostly affects older people. GHK-Cu is known to reverse some aging-related gene changes in cells, but it's a big maybe whether that translates into real protection of optic nerves in aging eyes. Direct evidence for GHK-Cu and eye health. To date, there is no direct research on GHK-Cu in glaucoma patients or models. We found no clinical trials or case reports of GHK-Cu being tested to lower eye pressure or protect the optic nerve. In animal glaucoma studies, few peptides besides the classic neurotransmitters have been tried. GHK-Cu has not been included. At the cellular level, there are no published experiments on GHK-Cu effects in retinal ganglion cells, trabecular meshwork cells, or optic nerve tissue. By contrast, some other peptides have been tested. For example, one study showed that a peptide named peptane 1, given by injection, could enter the eye and reduce RGC death in rats. This proves peptides can reach retinal neurons when administered properly. However, peptane 1 is unrelated to GHK. We have no data confirming that GHK-Cu crosses into the eye or alters eye nerves. Any suggestion that it might work in glaucoma is purely hypothetical at this point. What would need to be true for GHK-Cu to help glaucoma? For GHK-Cu to have any real benefit in glaucoma, several conditions would have to be met. Effective delivery. GHK-Cu would need to reach retinal ganglion cells or trabecular meshwork cells in sufficient dose. Because GHK-Cu is a peptide, it may not easily penetrate tissues. Intravenous or topical routes must actually deliver active GHK-Cu into the eye, for example via special eye drops or injection. Cellular effect. Once there, GHK-Cu would need to meaningfully reduce RGC vulnerability. This means it should decrease oxidative stress or inflammation in those cells or boost their mitochondrial health. It should stimulate repair pathways in the retina without causing unwanted fibrosis. Correct copper balance. Any copper delivered by GHK-Cu must be safely managed. This assumes the person's copper metabolism is normal, liver function, zinc levels, etc. Too much free copper could worsen oxidative damage, so GHK-Cu would need to supply copper only as needed and not overload the system. No interference. GHK-Cu must not cancel the effect of standard glaucoma medications or cause eye irritation. It should be safe with prostaglandins, beta blockers, or surgeries patients already use. Proven benefit beyond pressure. Importantly, a study would have to show GHK-Cu provides extra protection that is not just due to any unseen pressure drop. In other words, it should slow visual field loss or RGC death, even in well-treated patients. Volume and dosing. The correct dose and schedule would have to be found. Over long periods, would the body adapt or clear GHK-Cu quickly? Could injections be needed? These questions must be answered. These are just examples of needed truths. Currently, none of these conditions are validated for GHK-Cu in glaucoma. Possible risks and unknowns. Using GHK-Cu systemically or in the eye carries uncertainties. Unproven safety in eye. While GHK-Cu is generally safe on skin, its effects inside or near the eye are unknown. There might be inflammation or toxicity if given incorrectly. Metal imbalance. As mentioned, chronic GHK-Cu use could perturb copper and zinc levels. Some supplement peptides are not pure or standardized, leading to unpredictable copper loading. Quality control. Many GHK-Cu products exist as research or cosmetic-grade ingredients. They vary in purity and dosage. 98% purity is often claimed. Impure or mislabeled product could have side effects or no effect. Therapeutic delay. Perhaps most important, using GHK-Cu without evidence might delay proven treatments. Glaucoma therapy, eye drops, surgeries, etc. should never be substituted with an unvalidated peptide. Unknown interactions. GHK-Cu has broad gene effects. In theory, it could interact with other medications or carry off-target effects that we do not yet know. Because GHK-Cu studies have centered on skin and wound healing, we lack data on its long-term systemic use. Without careful trials, unexpected issues might arise. Conclusion: GHK-Cu is a fascinating molecule with wide-ranging repair and antioxidant actions in the body. Many of the processes it influences, redox balance, inflammation, collagen production, aging genes, overlap with pathways implicated in glaucoma. This overlap suggests a biologically plausible connection. For example, both glaucoma and GHK-Cu involve oxidative stress, copper biology, and aging. But crucially, plausibility is not proof. There is a complete lack of direct evidence that GHK-Cu benefits RGCs or lowers glaucoma risk. All we have are hints from other tissues. At present, the GHK-Cu glaucoma link remains speculative. We can say GHK-Cu theoretically could target some glaucoma-related pathways, but we cannot say it treats or prevents glaucoma. Glaucoma patients should rely on established treatments, eye pressure control, regular monitoring, and discuss new ideas with their ophthalmologist. If GHK-Cu is ever tested in glaucoma trials, scientists will look for evidence that it truly protects optic nerves beyond what pressure lowering does. Until then, claims about GHK-Cu for glaucoma should be taken with caution. Plain language summary. In short, GHK-Cu touches on many processes, like oxidative stress and repair, that occur in glaucoma. That makes it biology plausible as a helper peptide. But right now, it's only a theory. We have no solid proof it does anything for glaucoma, so it must be studied formally. And until then, glaucoma care stays focused on lowering eye pressure and watching the optic nerve. Biological pathway, why it matters in glaucoma, how GHK-Cu might relate, evidence strength, main caution. GHK-Cu upregulates genes and quenches radicals. Oxidative stress. RGCs die from cumulative oxidative damage to proteins' lipids. GHK-Cu upregulates antioxidant genes, SOD, HO1, etc., and quenches radicals. Moderate lab data. Antioxidants haven't proven to stop glaucoma in humans. Mitochondrial function. RGCs have high energy needs. Dysfunction triggers cell death. GHK-Cu carries copper, a cofactor for cytochrome oxidase, but no direct proof. Weak theoretical. No data that GHC-Cu enters RGC mitochondria or boosts ATP. Neuroinflammation. Chronic microglial astrocyte activation damages RGCs. GHK-Cu has systemic anti-inflammatory effects in wounds. Genes are modulated. Weak. No eye data. CNS inflammation in glaucoma is complex. GHK-Cu's effect unknown. Copper homeostasis. Copper is cofactor for protective enzymes, SOD, but toxic if free. GHK-Cu supplies copper and can bind free iron, possibly helpful if balanced. Speculative. Excess copper can cause oxidative harm, balance with zinc needed. Connective tissue ECM. Abnormal collagen ECM accumulation stiffens drainage tissue, raising IOP. GHK-Cu strongly stimulates collagen and ECM remodeling in wounds. Speculative. Unclear if remodeling meshwork would lower or raise IOP. Vascular regulation, poor optic nerve blood flow, SP in normal tension glaucoma, harms RGCs. GHK-Cu promotes angiogenesis in wounds, which could suggest improved microcirculation. Speculative. No proof it changes ocular blood flow, could cause aberrant vessels. Aging and repair signaling. Glaucoma risk rises steeply with age. Impaired repair damage. GHK-Cu can reset age gene profiles and stimulate repair genes. Speculative. Shifting skin gene expression may not translate to nerve protection. Important. This article is for research and education only. Glaucoma treatment must remain supervised by an eye specialist, focusing on proven strategies like IOP control and vision monitoring. GHK-Cu is not a substitute for those treatments. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.